Hey, Nate.
Yes, Sam.
You know how everybody these days is getting like their personal genome sequenced?
Yeah, I mean, there's tons of companies doing that now.
Yeah, I thought like back in, I don't know, like almost 20 years ago, they sequenced the human genome.
But if there is a human genome, why are we all so different?
I don't know.
Hey, I got a question about that.
Hello, and welcome to another episode of Hey, I Got a Question About That. I'm Sam.
I'm Nate, and this is a podcast and video series where we talk about all the fascinating research going on here at the Penn State Eberly College of Science.
On this episode, we're joined by Santhosh Girirajan. He's an associate professor of biochemistry and molecular biology and of anthropology here at Penn State, and he studies genetic variation, particularly in relationship to neurodevelopmental disease. And we talked to him about the human genome and if there is a human genome.
Right. There was a lot of good information in this talk, so let's just get right into it.
So we are joined here in the studio today by Santhosh Girirajan. He's an associate professor of biochemistry and molecular biology, as well as anthropology here at Penn State. Thanks for joining us, Santosh.
Thanks, Sam. Thanks, Nate.
Can you tell us a little bit about your research kind of generally?
Yes, I'm a human geneticist, and we study the genetics, complex genetics of neurodevelopmental disorders, such as autism, intellectual disability, schizophrenia, and epilepsy. So basically, we use a series of different types of experiments in Drosophila flies. We work with families with affected children, and also we develop new bioinformatics tools to analyze data.
So Nate and I were talking a little bit, one of our previous episodes, we talked about the Nittany lion genome, and something that came up was kind of the human genome. And I was wondering, since you study human genetics, what is kind of the human genome?
So there is no the human genome because what we know so far is that on an average we have about 3 billion base pairs, which we call the human genome. And every person differs in the length of their genomes and their composition of their genomes. So the first human genome project was kind of a first step forward in understanding what human genomes contain. But in reality, what we know so far is there's a lot of variation between genomes. So there is no the human genome. There are multiple different types of human genomes.
So when you say there's variation in the genome, what does that mean exactly?
So in principle, most of us, or all of us, should contain so many number of genes and a certain number of base pairs and what we call regions, DNA between genes, intergenic regions, introns, exons, and repeat elements and everything. But there are, what we've come to understand in the past 15 years is that certain regions in the human genome can be deleted or duplicated or can exist in multiple copies in different individuals. So this can vary dependent on the ethnic group, the population where they come from, and also if among individuals who have a certain type of neurodevelopmental disorders, you can find these deletions and duplications. So which means that my genome can be longer than your genome, and I could contain certain basis DNA elements that could be different from yours.
So if my genome is longer than yours, is that a good thing?
There's nothing like good or bad. All these variations actually confer certain traits, such as height, weight, and also certain quantitative traits, such as the level of sugar in your blood, and also some of these traits can be good in terms of, for example, people have implicated, for example, multiple different types of variation to cognitive ability. And at the same time, people have identified specific changes in the genome contributing to increase in cholesterol levels, contributing to heart disease. So they're all traits, and I would say they all fall within a distribution. Some people have less, and some people have more, or if it's a quantitative trait. So that's what makes us different. So diversity that we see in the population, not just in humans, but in any kind of organisms, you would see the variation in the genomes contribute to their the variability that you see in their traits or phenotypes.
But So Santosh, when the Human Genome Project kind of completed, wasn't the idea that we could use that to compare and help us like identify regions in the genome or genes that are disease-causing? So it's kind of seems like where we vary from that human genome, those things might be, cause problems, might be disease-causing.
Right. So there's the variation or the differences in our traits can explain a lot of things in the sense we can say, well, this person is short, this person is tall, but the extreme of it can be pathological. For example, you can have individuals who are, you can have a group of individuals who fall within a distribution for weight. But if you see somebody who's at the extreme of that distribution, you would see that that particular variation is actually contributing to that extreme variation, and potentially that's pathological. So basically, the disorders that when we started out, for example, in the Human Genome Project, the initial expectation was we would find something like a Mendelian, which is a straightforward single genes contributing to a single phenotype, a single disease. What we've come to understand now is most disorders are complex.
So when you say Mendelian, you're talking about Gregor Mendel.
Gregor Mendel,
who's
yes.
This, 19th century botanist who studied peas, and he found these really simple traits. So a pea would be either green or yellow.
Yellow, yeah. He was talking about dominant traits or recessive traits. You have 2 copies of something called a bad allele, then you're affected. It's a recessive. Or if you have 1 bad allele, then you're affected, which is a dominant trait. But people, and this is basically for most of these Mendelian traits where that's what we thought. Most of this is going to be Mendelian traits. We're going to find single genes or individual genes contributing to it. But over time, we've realized even those quote-unquote Mendelian disorders are actually modified or modulated by multiple different other genes, which is in the genetic background.
So when you talk about traits that are kind of continuous and that people fall within some distribution, since one of the things you study is autism.
Correct.
I guess I don't know what the most up-to-date terminology is, but I know that people talked a long time about autism spectrum disorders.
Correct.
Is there a place along that spectrum where you kind of go from someone who has a disorder to someone who doesn't have a disorder?
Right. So autism is an interesting, a very complex disorder. It has multiple dimensions. So the way people talk about it as a spectrum is actually individuals with autism or affected with autism have defects in multiple different their distributions within multiple domains, which could be cognitive or psychiatric or psychological or behavioral domains, or and also some of these could be metabolic domains. So each one of this, if you take into account each one of them, every child diagnosed with autism will have different, will be affected differently in each one of these domains. And if you put all of these complex phenotypes together, it's really hard to really diagnose a set of individuals to have one particular disorder. So that is where the complexity comes in. And added to that, what I would say is that autism, in terms of just diagnosis by a clinician, it's not the current diagnostic techniques that we have. It's not sensitive enough or completely sensitive to identify all such domains. So you have to have more specialized ways of looking at it. And we are looking at them in a more looking at a few domains and trying to diagnose autism. That's why people have been now doing imaging studies and all other, even including genomic studies and so on.
So is autism a Mendelian disorder?
No. So autism, like I mentioned before, from the Human Genome Project, we expected many of these disorders are going to be Mendelian, which means that single genes can be identified as causative for these disorders. But what we come to recognize is that disorders such as autism, which is extremely complex, containing multiple different dimensions in terms of the associated phenotypes, are caused by multiple genes. And in fact, from what we know, where we've sequenced, people have sequenced more than, I'd say, 50,000 individuals with autism, and we know that there are more than a few thousands of genes. So it's either single gene, some single genes cause a specific subtype of autism, or combinations of genes contributing to a more complex form of autism.
How do you kind of deal with this complexity in your research?
So this is the biggest challenge that we all face in our field. The aspect of dissecting the autism into subtypes and trying to deal with individual subtypes has been the recent developments in the field. What people do is we are trying to dissect. For example, what we do in my lab is dissect autism based on some of the comorbid features. Comorbid features that go along with the typical triad of autism features, where people have repetitive behavior, speech delay, and decreased social abilities. So the comorbid features are intellectual disability. For example, these individuals, some of these individuals with autism have IQ scores less than 70. They have cognitive defects, and some of them have epilepsy. Interestingly, individuals who have epilepsy, there's an interesting aspect about autism is that the autism as such, it's highly prevalent in males compared to females. On the other hand, if you find individuals who have autism plus epilepsy, you would find more females, in the sense that particular ratio kind of balances out. So you would see that there are more females with autism and epilepsy, and that's an interesting aspect. So we are trying to dissect individual components of autism and trying to see if we can find certain patterns or certain combinations of mutations in genes or certain genes that could contribute to these subtypes. So it's a Herculean task. It's not going to be so simple, because every family we've seen with autism is different from another family that we've seen with autism. So people, this is it's a kind of a often said sentence is that if you've seen a child with autism, you've seen one child with autism. That's what it is. So it is true.
So Santosh, you're talking about how autism is this complex disease and probably has a lot of different causes in the genome. Are those variations that you see in people with autism also seen in other people who haven't been diagnosed with autism?
So that's a good question. So there have been, you can see both sides to it. So for example, there are rare causes for autism or autism type disorders that are extremely rare that you typically don't see in the general, what you call general population. Then there are other variants that you see in the general floating around in the general population, contributing to very, very subtle clinical phenotypes or clinical features that manifests into autism when you have other combinations or other mutations in the genomes. So this is very interesting because we all have some level of susceptibility for certain things. For example, you could have susceptibility for certain traits, which I would say susceptibility not necessarily in a bad context. For example, we carry alleles, what we call variations of a particular gene, for example. Can, for example, for height, for weight, and things like that, for, like I said, blood glucose level and things like that. And similarly, for we have certain predilections for certain disorders. We carry some of these, and it's just a matter of the context. You have a certain variation, you pass this a certain variation on to the a child, and the child also inherits another variation from your spouse. So that combination probably can lead to manifestation of that particular trait that was hidden. So, or can be the trait that would have led to very subtle changes, or certain subtle traits in you could really manifest as a more severe one in the child. So in fact, some of these, for example, like I mentioned, for example, the social attributes, your social behavior, if measured, is actually would fall into a good distribution. And some of us who are at the kind of not so extreme, but more towards the tails, maybe we do carry some of those mutations or genetic changes that lead to these social changes. And this, when combination with others, potentially leads to more severe forms of social defects and things like that.
So if I would get my genome sequenced, what are some of the things that I could find out?
So the knowledge in the field, especially in genetics, is that we have been able to attribute certain genetic changes to certain traits, certain disorders, and certain, and also can identify the population or your origins, for example, where exactly in which part of the world did your ancestors came from, and things like that. So you can certainly, for example, you can find out if you have a risk, not necessarily you, but individuals can find out if they have risk for example, breast cancer, for risk for Parkinson's disease. But they're again associated. They come with, they tell you how much risk you carry. They won't tell you whether you will get it or not get it. At the same time, there are certain genetic changes if you find out if you carry it. For example, certain deletions and duplications in the human genome that have been seen in children in children who have neurodevelopmental disorders. Several of those have actually been inherited from an apparently healthy parent or parent who has subtle features. So it will help in actually providing some genetic counseling, or if we can actually start rehabilitation at an earlier stage so that we can reduce the severity of symptoms in the child. Yes.
So would you get your genome sequenced?
It's a good question. So would anybody would like to get their genome sequenced? Well, first of all, it's like a kind of a very personal choice. In my case, I would say the way I see the human genome is I would really like to get my genome sequenced. But would I want to analyze my genome to know what's going on? Well, I would analyze it. It's like the way I see it is I would like to carry my genome in a pen drive and go to the doctor every time I have some issue. Like if I find for example, there's a family history of hypercholesterolemia in my family, and I want to know if I have mutations in some of those hypercholesterolemia associated genes. At the same time, if there's something that I come to know, if I get some new information in terms of genetic or clinical features just by regular physical tests, then I'd go back to know what's going on. But I wouldn't go back to ask, would I get this in the future without any symptoms happening to me? No, I wouldn't do that. That's me, but no, that's I think the way we should understand the genome is it should be more iterative. You should go back and forth to doctor, and you should add more information that becomes available through research and rigorous analysis. Actually, we have the data that we have currently for some of the disorders are based on a few sample sizes. So we have to have robust analysis to understand the real contribution of these variants to diseases.
So you've talked a lot about how there's this variation in the human genome, and we kind of all have different versions of the genome. Is there something that you would call normal, like the normal human genome?
This is a very it's a tricky question, very hard to answer. The reason being that the genomes that we use today, we call this as a reference genome because the genome that we use to compare in our analysis, in laboratories or in clinical diagnostic settings, is a reference genome which we think is, quote unquote, a normal genome. We think that this genome should contain everything, and the DNA is all, there are no changes, mutations, and every mutation is actually apparently normal. So the best way to think about this is in family studies that we perform, we compare the affected child's genome to the genomes of the parents, because parents typically are not affected. We look at simplex families where the child is the only one who's affected. So that is where the comparison becomes. So whenever you talk about normal genomes, it's based on a context. If you're looking at individuals with a particular trait, your comparison set should not contain that trait. But they might contain something else. They might have something else going on. So it becomes very contextual, but there's no way to, I don't think there is anything called a normal genome, because everybody has their own predispositions for certain disorders or susceptibility for certain traits. We are actually a consequence of the genetic changes and the environmental factors and all the evolutionary changes that happened in our past few generations. So we are the consequence of all of those things. So there's nothing called normal now.
So where do you see the future of genomics heading? Am I going to be walking into the office with my genome on a pen drive?
Possibly. I think there are a few things that this particular field is heading towards. One is I think that this is becoming more population scale, which means that everybody in the population is going to get some kind of a genome sequencing done. Either you're going to get sequenced for a selected number of genes, or all the genes in the genome, or everything in the genome. And the other aspect, I think, is that all the physicians should get educated and informed in terms of genomics, and also the clinical diagnosis should also involve genetic diagnosis. So I think we will, at some point, we will all be owning our own genomes. You will be carrying it with you, and the biggest question that people have been asking now is, who owns our data? Who owns our genetic data? So the individual, at the individual level, you own it, but you can still use it to understand what it means. And I would say that again, you have to have an indication to go back to your genome. If you're really looking for something, if you want to know where your great-grandparents came from or grandfather came from, which part of Europe or which part of the world, you can use your genome. And if you want to know if you have predisposition to, or if you will be affected with something because based on some family history, you can go back to your genomes.
Well, that was really, really interesting. Thanks so much for joining us, Santosh.
Yeah, my pleasure. Thank you very much.
Thank you.
That was great.
Yeah, I always knew that there was differences in the human genome, but I never realized that there could be differences in the length of the human genome too.
And I thought it was a really good point that Santosh made, that even though there are these differences, it doesn't necessarily mean, one is good, one is bad. There's really no value judgments. It's just the differences that make each of us unique.
So if you want to learn more about Santosh's research, we'll have links to his work in the show notes below. And we'll also have links to the Eberly College of Science website, where you can find information on other research going on in the college.
So thanks for joining us on this episode of Hey, I Got a Question About That. And if you haven't already, check out our other episodes, which are available wherever you get your podcasts, and also in video format on YouTube. And don't forget to like, subscribe, and leave us a comment.
Europa moments.
What do we talk about?
This is a podcast and video series where we talk. I should have looked at the camera. I'm sorry.
Hello.
Hey!
Human genome, and what would... why do I try to keep going? Did I manage to not say cool the entire time?
It's really neat to see. So if you want to learn more about Santosh's research... Nope. Santosh's research. I need to slow down when I speak. So if you want to learn more about... Nope. I'm going to get in my head again. It's going to be a Europa moment.
Subscribe that smash button.
Beat that subscribe button into submission.
We'll see you next time.
Thanks for tuning in. We'll see you next time. We'll be like LeVar Burton. I'll see you next time. Didn't you watch Reading Rainbow?
I did. I just remember the song. Take a book.
It's in a book. Reading Rainbow.